Hello friends and dear children. Today I am going to discuss some questions related to plant cell culture and its application. So first of all, try to know the question related to antisense RNA technology. In this case, we are going to silence the function of the particular gene through this antisense RNA technology. Here example is flavor saver variety of tomato. A flavor saver variety of tomato is produced by which technique? Options: genetic male sterility by Barnage bar star systems, antisense RNA technology, DNA microarray technology, molecular farming. So this flavor saver variety is going to produced by a molecular technique, that is antisense RNA technology. So correct answer is antisense RNA technology. Such type of question are coming in so many competitive exam related to biology and biotechnology. Next questions. Some question also asked related to image or photographs. So try to identify the following structure or diagram related to plant tissue culture. First choice is, try to observe clearly. First choice is Callus, second is Protoplast, third is Somatic embryo, and fourth is artificial seed. So the correct answer, can you predict? If you will not predict, the answer is Callus. Okay. Next questions related to diagram itself. Identify the following structure in a given diagram related to plant tissue cultures. So the plant tissue culture is related for the production of hybrids, genetic study, gene manipulation. If you are doing such activity, then if you have no any cell wall in any plant cell, then it is easy to transfer the gene, because cell wall create a hindrance for transferring the gene inside the plant. So if you observe the choice, this diagram is related to what? Somatic embryo, correct or not? Callus, explant, protoplast. You know, protoplast means cell without cell wall. If you observe minutely, then you can observe clearly that there is no cell wall. Okay, so the correct answer will be protoplast culture. You can easily transfer any gene, you can easily produce any useful biomolecules inside this protoplast cell through protoplast culture. Next questions. Number of the foreign gene has been transferred in transgenic variety of golden RNA. Have you heard about golden RNAs? Or a rice variety that contain beta carotene gene and iron gene. Beta carotene is immediate precursor of vitamin A. So if there is sufficient amount of vitamin A in your diet, no chance of night blindness. Your eye will work properly due to excess sufficient amount of beta carotene compound or vitamin A compound present in your body. Next, that is iron deficiency. Due to deficiency of iron, people going to suffer from anemia. So if such variety will taken by the poor people or children of the poor people, they will not suffer from such type of disease like anemia and night blindness. So scientists had created a new variety, that is golden iron rice, by transferring 3 genes: PSY gene, CRT1 gene and CRT2 gene. So how many number of genes can you predict? 1, 2, 3, 4. So that is correct answer is 3. Next, tearless onion is produced by a molecular techniques just like antisense RNA technology. One technique is similar to antisense RNA technology, that is gene silencing. If you are going to silence the functioning of particular gene, suppose you are cutting the onion and you observe your eye feeling tear. So somehow some character or gene is responsible for that. If you are going to silence, means not functioning of that gene, then if you have created such transgenic plant of onion and when you are using for your vegetable purpose, no chance of production of tear. So we get tearless variety of what? Onions. So we can do such things by gene silencing, means creation of transgenic plant of the onion through gene silencing. So just go through the choice: gene silencing, first choice; Barnage bar star system, second choice; western blotting, third choice; northern blotting, fourth choice. So correct answer will be gene silencing. Yes. Next question. It is also related to diagrams. So nowadays in so many competitive exam, diagram related question is going to ask, and even in CBSE also this type of question is going to ask by CBSE. So identify the following structure in given diagram related to plant tissue culture. So you have to minutely observe such diagram. It is callus or protoplast. Callus, undifferentiated growth, unorganized growth of the plant cell. We cannot say it is root part or a stem part of the plant in initial stage of plant tissue cultures. So correct answer, it may be or may not be protoplast. Cell without cell wall, such type of structure is not you are going to observe in the above diagram. Somatic embryo or that is the third choice, and explant. Explant, that is a small section of the apical meristems, that is explant. But such type of structure you are not going to predict through this diagram. So correct answer will be somatic embryo. This somatic embryo will derive from callus, and from where callus is going to derive? From explant. So this diagram is completely somatic embryo structures. Okay. Next, this is also question related to diagrams. Identify the figures in the following diagrams. Okay. So round of ready variety of soybeans. Okay. Yes. Non-transgenic and transgenic cottons, transgenic papaya plant, golden iron rice. Okay. So if you observe, I am asking to identify the diagram in B. If you observe the ball, so ball is generally present in case of cottons. Okay. So you know BT cotton. BT cotton is due to presence of BT gene that scientists have transferred from Bacillus thuringiensis bacteria to uh, normal cotton plants by gene transfer techniques. So that plant is going to produce BT crystal protein, although this BT crystal protein is soluble in the cell sap of that plant, but when pink bollworm or cotton bollworm will suck that liquid, then this liquid is going to convert into solid crystal, and this solid crystal kill the bollworm or pink bollworm or cotton bollworm. So correct answer will be the transgenic and non-transgenic cottons. In the you can observe one part of this B diagram is non-transgenic and other part is transgenic. Just for control, we are the figure described as transgenic and non-transgenic. Okay. So in the left side you can observe the non-transgenic, in the right side that is transgenic. Okay. Next questions. It is also related to diagrams. So can you predict as a biologist or as a biotechnologist or if you are a study in the field of biotechnology, biology or biochemistry, then try to guess that diagram what it can be. It is what? It is somatic embryo? No. It is explant? No. It is callus? No. It is artificial seed. So artificial seed, that is encapsulation of calcium alginates. Encapsulation of calcium alginates. This calcium alginate is act as desiccating agents. You can observe the A layer is calcium alginate, B is somatic embryo and C is endosperm. So through this artificial seed we can rapidly multiply the seedlings of any plant varieties in adverse environmental condition as per the demand of farmers. Okay. So correct answer will be artificial seed. Next, identify the technique of plant tissue culture in the following diagram A and B. If you are a biologist, biotechnologist, biochemist or plant breeder or scientist, then you can easily identify. So this is a plant of banana. So banana is going to multiply through which technique? There is so many techniques for multiplication of the plant through plant tissue culture. One is somatic embryogenesis, one is organogenesis and one is micropropagation. In case of somatic embryogenesis, we are going to multiply the plant from explant to callus and callus to stem portion and further in for the rooting purpose. But in case of micropropagation, we are going to bypass the one particular stage, that is callus. So in this diagram there is no formation of callus and we can multiply the plant without formation of callus into so many multiple shoot formation. You can observe clearly in this diagram. So try to observe the choice of your questions, that is micropropagation and multiple shoot formation acclimatization in greenhouse. So right side, that is the greenhouse, and in the left side we are going to proliferate the plant numbers through proliferating the stem formation, and further we segregate it after root development. So artificial seed formation, it is wrong. Protoplast culture and pathogenesis, it is wrong. Organogenesis, hybridization, it is wrong. Okay, so correct answer will be micro propagation and multiple shoot formation, acclimatization in greenhouse. Okay, next, that is the source of agar. It is source of agarose is agarose used as a solidifying agent. Yes. So generally this agarose is polysaccharides, and we are getting this agarose from particular algae. So can you guess from which algae it is coming? It is a very fine powder used as a solidifying agent just to grow any microbial cell or any plant cell in solid surface media. If you are going to prepare a media, there is two type of media. One is liquid media, that is known as broth, but if it is solid media, then you must add agarose. Okay, you can also use agar or agarose as a solidifying agent. So generally agarose used for separation of DNA, but agar act as a solidifying agent just to grow of the plant from the explant, or from the you are going to multiply the bacteria on the surface of agar. Okay, so agar and both are deriving from the same algae. As you can observe, red algae, blue green algae, brown algae, orange algae. Can you predict? Correct answer is red algae. So thank you very much. You try to learn all these questions related to your coming exam in CBSE or in any other competitive exam. Thank you and have a nice day.